Hi everyone, this is chapter 11 on characterizing and classifying prokaryotes. So let's talk about the general characteristics of prokaryotic organisms. We'll start with looking at the morphology of them. This is the most diverse group of cellular microbes that we talk about. They thrive in a variety of habitats. There's only a few that are capable of colonizing humans and causing disease. So there's a lot more bacteria out there that are non-pathogenic to you and I or to other animals. They exist in a variety of shapes, and that's what the term morphology means, the shape. So we'll start with this, and I think we looked at this a little bit in lab already, but just as an add-on to that and some additional information besides the basic lab information. So our typical prokaryotic morphologies include coccus, which is round, cocobacillus, which is a combination between a coccus and a bacillus. Bacillus is rod-shaped, so you can see that it's kind of so more of like an oval shaped, but not quite as ovally as a bacillus, if that's possible, and then a vibrio, which is like a bacillus with a curve to it. Spirillum, which is spiraled, and then we have a true spirochete. So you see all of the twists there. There are some that are pleomorphic, meaning that they change, and then sometimes they cluster together and make what looks like a star. Endospores, I think we've talked about this a little bit in the staining lab, but let's talk about them a little bit more. Endospores are produced by gram-positive bacteria, specifically in the Bacillus and the Clostridium groups. Each vegetative cell transforms into one endospore. So a vegetative cell is a cell that's just doing its everyday job, just minding its business, doing what it normally does. And if the conditions become unfavorable, that vegetative cell transforms itself into an endospore. This is a defense strategy against these unfavorable conditions. So maybe it's too hot, or it's too cold, or it's too dry, or it's too wet. There's 1,000 different scenarios out there, but if it's not the proper environment for that particular vegetative cell to do its everyday job, it has the ability to transform into an endospore. Not all cells can do this. Not all bacteria can do this. Again, this is just in Bacillus and Clostridium that do this. All right. Um, this is a concern to food processors, healthcare professionals, and to governments, because food processors, if it's an unfavorable condition, the bacterium could transform itself into a endospore in your food. Clostridium is a classic example of this because it can cause botulism. Healthcare, same kind of thing. There are some Bacillus and Clostridium we absolutely positively don't want to turn into endospore because they can infect our patients, and governments because unfortunately these can be used as weapons. They can be weaponized. So you saw this in the slides in lab. We talked about the location of the endospores. So you'll see what looks like on the left-hand side, what looks like a glow-in-the-dark dot. That's just the type of staining that they've used. And so you can see the endospore. The vegetative cell is the one that they're pointing to that doesn't have one of those. That's just an everyday bacillus doing its regular job. And then you've got some that have sporulated. They've gone into this sort of suspended animation where they're dormant. On the right-hand side, they're showing you a different staining technique with different bacteria, and they almost look like they have a little loop because the endospore is at the very end of the bacterium. Let's talk about how prokaryotic cells reproduce. So they all reproduce asexually, and there's three main methods that they use. The most common of those is binary fission, there's snapping division, and budding. All right, a little bit more about binary fission. Basically, the cell is going to make a copy of its DNA, and then some extra materials, cytoplasm, etc. And then it's going to pinch itself into two. That's all there is to it. So there's nothing exchanging. There's nothing real complicated about this at all. And because it is so uncomplicated, it can happen as fast as every 30 minutes. Compare that to a eukaryotic cell when it goes through mitosis. The process of mitosis takes about an hour, and how frequently that cell is going to mitose can be years between events. Or months or days, but in binary fission, this can happen very, very rapidly. Snapping division is a variation of binary fission. You have an older portion of the cell wall, and you have a newer portion, and it literally just ruptures and snaps itself in half. Budding is another way that these cells can reproduce. So in this particular case, the DNA is attached to the cytoplasmic membrane, is replicated. The cell is going to elongate and forms what's called a bud. And then that young bud is just literally going to snap off. So again, not anything especially glamorous. It can happen very rapidly. So a little bit more to talk about. A certain group of bacterium, Epulopisculum, and its relatives have a unique way of reproducing. The live offspring emerge from the body of the dead mother cell in what's called viviparity. This was first noted in the prokaryotic world. And so there's what it looks like. So you can see the live offspring that are inside of the mother cell, the mother bacterium, and the mother bacterium has died. And then these are going to erupt from that and go out and do what bacteria do. So we talked about the shapes, and we talked about how they reproduce. We can also talk about how they are arranged. Sometimes they are just a solo bacterium by themselves, but often they are in arrangements. And so when we look at coci, the round ones, there's a lot of different ways they can arrange themselves. So they can be a diplococi, meaning there's two of them that are connected together. They can be a streptococi, which means they're a whole string. They can be formed in a tetrad, which is four of them, or a Sarkini, which is, I think, 2, 4, 6, 8 of them, if I remember correctly. Or they can be in a Staphylococcus, which looks like a grape cluster. Bacilli, again, they have fewer ways that they can arrange themselves, but they can arrange themselves. Sometimes they're just a single, like we see in the top picture there. They can be a diplobacilli or a strepto. Strep always means a chain of them. And they can also form themselves into pallicides. And when they do in pallicides are kind of cool because they can make lots of different shapes. Like they can be a V shape, or they can make almost look like different letters of the alphabet. So switching gears, let's talk about modern prokaryotic classification. So currently, our classifications are based on genetic relatedness of the ribosomal RNA sequences. There's three big domains that we talk about. There's archaea, there's, or some people say archaea, bacteria, and eukarya. All right, we'll start with talking about archaea just a little bit. Their common features are that they lack a true peptoglycan molecule in the wall. The cell membrane lipids have branched hydrocarbon chains, and the AUG codon that it uses in replication is going to code for methionine. There are two phyla in archaea. There's the Crinoarchaeota and the Euroarchaeota. These all reproduce by binary fission, budding, or fragmentation. Fragmentation just is kind of like budding, where just a piece just breaks off and makes uh, a new structure. They can be coci, bacillus, spirals, or pleomorphic. They are not known to cause any disease, so we're going to cover them in here because that's what makes micro comprehensive. But you'll never ever see any of this out in your job as a healthcare provider. There's some uh, transmission electron micrographs of what they look like. They're fun to talk about though because they live in weird conditions, strange conditions, unusual conditions, and so they're kind of fun to talk about in that particular way. So we'll run through these real quick. The extremophiles, these require extreme conditions to survive, either in temperature or pH or the salinity. The prominent members of this are the thermophiles and the haliophiles. So the thermophiles, the DNA, RNA, cytoplasmic membranes, and proteins don't function properly below 45 degrees Celsius. So again, this is Celsius, not Fahrenheit. So these like it pretty hot, right? Hyperthermophils require temperatures over 80 degrees Celsius. There's two representative genera here. There's a Thermococcus and the Pyrodictium. So where will you see some of these? Like in the hot springs. So if you go go out west to the Hot Springs National Park, you can see archaea, example of archaea living there. I think they live in Arkansas as well. You may have some hot springs up where you are where you have some hyperthermophilic archaea living. Haliophiles like things really salty. All right, so that's a different way to be for sure. They depend on greater than 9% sodium chloride to maintain the integrity of their cell wall. They may contain red or orange pigments. They may protect from sunlight. And the most studied of these is Haliobacterium celarium. So here is a picture where there's highly salty water, and you can see the haliophiles, the red and the yellow colors that you see living there. The methanogens are the largest group of archaea. They convert carbon dioxide, hydrogen gas, and organic acids into methane gas. They convert organic waste in ponds, lake, and ocean sediments to methane. Some live in the colons of animals. One of the primary sources of environmental methane is through the methanogens that live in the colons of animals, and particularly ruminants, and especially cows. We hear about that. They have produced 10 trillion tons of methane that is buried in the mud or ocean floor. So, as some of you might know, I live in the southeast portion of the United States, and I have a lot of marshes around me. We don't call them swamps here, we call them marshes. And they are full of these methanogens, and you can smell them. There is a very distinctive smell in all of our marshes around here because of these. They call it pluff mud. It's spelled Pluff mud, and it's only something you smell along the low country of South Carolina, the coastal empire of Georgia, running along the area where I live. And it's a very distinct smell from these methanogens. Deep branching and phototropic bacteria, the deeply branching bacteria, the scientists believe these organisms are similar to our earliest bacteria. They are autotrophs, meaning that they make their own food from sun, and they live in habitats similar to those thought to exist on Earth in early Earth. Aquifex is one that we talk about. It's considered to be the earliest branch of bacteria, and Deinococcus has outer membrane that's similar to gram negatives, but actually stains gram positive. So phototrophs contain photosynthetic lamella. Most of them are autotrophic. They divide into 5 groups based on their pigments and the source of electrons for photosynthesis. We have the blue-green bacteria called the cyanobacteria. We have green sulfur bacteria, green non-sulfur bacteria, purple sulfur bacteria, and purple non-sulfur bacteria. The cyanobacteria are gram-negative phototropes. They're thought to have transformed an anaerobic atmosphere into the oxygen-containing atmosphere that we see now on Earth. Chloroplasts possibly developed from cyanobacteria, and some species reduce nitrogen gas via nitrogen fixation. And here's what they look like. If you get them underneath the microscope, they're green. Here is some purple sulfobacteria, named accordingly. And then here is just a chart. Again, archaea is not our focus in this course at all. I'm just briefly going over it so that you can have an appreciation for it. But other than asking you some very basic questions about archaea, I would not ask you this level of detail. All right, something that we do need to know about is the low G+C gram-positive bacteria. And we'll start with talking about Clostridia. These are rod-shaped obligate anaerobes, meaning they cannot be in oxygen. They form endospores, and we've already talked about why that is important in medicine and in industry. Clostridiums are microbes that are related to Clostridium include Apulopisculum, sulfate-reducing microbes, and Selenomonas. Another low G plus C gram-positive bacteria that we want to know about are the mycoplasms. These are facultative or obligate anaerobes. They lack a cell wall. These are the smallest of the free-living cells, and they tend to colonize mucous membranes of the respiratory and urinary tract. Here is a light micrograph of mycoplasm colonies. They look like a fried egg. All right, a few other low G plus C gram-positive bacilli and foci to be aware of. Bacillus, these are commonly found in the soil. They form endospores. Bacillus thuringiensis is what they call Bt toxin. It's used by farmers and gardeners as an insecticide, and Bacillus anthracis, which causes anthrax. So crystals of the Bt toxin that are produced by that particular bacteria. Some others, listeria. Contaminated milk and meat products is where you'll find listeria. They are capable of reproducing under refrigeration, and they can cross the placenta in pregnant women, which is one of the reasons why you have to be really careful about raw milk. Lactobacillus grows in the body but rarely causes disease. It's used in the production of various foods. Streptococcus and Enterococcus, these cause numerous diseases. There are various strains of multi-drug resistant streptococci out there. Staphylococcus is one of the more common inhabitants of humans. It produces toxins and enzymes that contribute to the disease. So here is a nice little chart of the characteristics of selective gram-positive bacteria that you might want to take a look at, specifically the ones that I talked about, the Clostridium, the Mycoplasma, the Bacillus listeria, and Lactobacillus. Page 2 of this chart has the Streptococcus and the Staphylococcus on it, and then the actinobacteria that I'm about to talk about. Okay, Corneobacterium, these are pleomorphic aerobes and facultative anaerobes. They produce metachromatic granules. The Mycobacterium, these are aerobic rods that sometimes form filaments. These are slow growing because they have mycolic acid in their cell walls, which is unique to them. And the Actinomycetes, these are form branching filaments that resemble fungi. There's some important genera of these that include Actinomyces, Nocardia, and Streptomyces. So here is a picture of acid-fast bacilli, or AFB. This is going to be the mycobacterium. Remember, we did in lab the acid-fast stain when you suspect that you have a mycobacterium that will not stain correctly using the gram stain technique. You use AFB. And then here is a light micrograph picture of a branching filament of the Acetomyces. Some gram-negative proteobacteria. These are the largest and most diverse group of bacteria. There's 6 major classes that we talk about: alpha proteobacteria, beta, gamma, delta, epsilon, and zeta proteobacteria. All right, looking at the class alpha proteobacteria, these are nitrogen fixers. So they're going to grow in association with the roots of plants. So typically this is important in agriculture. 2 important groups in there are going to be the Azospirillum and the Rhizobium. Additionally, hydrogen gas is produced by Rhodopseudomonas palustris, and that may actually be a potential biofuel in the future. And so there is what one looks like. Kind of looks like a banana. This is the nodules of a pea plant, and so you've got the bacterium that are in working in cooperation with the nodules, with the pea plant roots, and forming these nodules. The alpha proteobacteria, these are nitrifying bacteria. They result from the oxidation of nitrogenous compounds, and that's what's going to provide electrons. So this is important in environment and in agriculture. The group that we talk about here are called the nitrobacter. Purple non-sulfur phototropes, these grow at the bottoms of lakes and ponds. Some more in this particular class, the pathnogenic alpha proteobacteria that we would be more interested in. The Rixia, these are transmitted through the bite of an arthropod, so typically a tick or something in that group. They cause several human diseases. And Brucella, this causes brucellosis. A few other alpha proteobacteria, the Actobacter, the Gluconobacter, and the Caulobacter. Just showing you a slide of the growth and reproduction of this particular one. A plant gall. So if you know anything about plants, they can develop this structure called a gall, and it's a disease that happens to the plant. In the class beta proteobacteria, we have some pathogenic beta proteobacteria that we need to talk about here. Neisseria. Neisseria inhibits the mucous membranes of mammals and causes numerous diseases. Bordetella, which causes pertussis, and Burkholderia, which colonizes the moist environmental surfaces and respiratory passages of cystic fibrosis patients. A few more, the Theobacillus, this recycles sulfur in the environment. The Zugloia, which forms flocks that assist in the treatment of sewage. And then the last one that also impedes the flow of waste in treatment plants. So flocks are slimy, tangled masses of bacteria and organic matter. And there's what they look like. The gamma proteobacteria, this is the largest and most diverse group or class of the proteobacteria. We divide this into subgroups, including the purple sulfur bacteria, the intracellular pathogens, the methane oxidizers, the glycolytic facultative anaerobes, and the Pseudomonas. So here is a picture of the purple sulfur bacteria. So that is on the bottom. That is a roundworm that has been infected by purple sulfur bacteria. So some gamma proteobacteria, intracellular pathogens we want to be aware of. Legionella causes a disease called Legionnaire's disease. That's often associated with faulty air conditioning units. There's a long history to this, but it was first named because of a convention, I believe in Philadelphia, of Legionnaires that went there for their yearly convention, and there were um, faulty HVAC units, and it aerosolized the Legionella, and they breathed it in and got really sick from it. A whole bunch of them died. And then Coxiella, which causes Q fever. Methane oxidizers, these use methane as a carbon and energy source, and they inhabit anaerobic environments. The glycolytic facultative anaerobes, these catabolize carbohydrates by glycolysis and the pentose phosphate pathway, and we divide this into three families. Okay, so these are the three families of the glycolytic facultative anaerobes of the class Gamma proteobacteria. So we have the enterobacteria. These are straight rods, oxidase negative, which is a test you're about to learn how to do, and peritrichosis flagella, or they can be non-modal. Classic representative of this is E. coli or Escherichia. And then there's the enterobacteria, Serratia, Salmonella, Proteus, Shigella, Yersinia, and Klebsiella. They're all in this particular family. And then you have the Vibrioniaceae. This is the Vibrio, the curved shaped rod. These are oxidase positive, and they have a polar flagella. And this is the causative agent in cholera. And then the Pasteurellacea, which are the foci and straight rods. These are oxidase positive, non-nodal. Haemophilus, this causes a type of meningitis in children, middle ear infections, and pneumonia. Another one we want to talk about in here are the pseudomonads. These break down numerous organic materials. These are important pathogens of humans and animals. Pseudomonas causes urinary tract, ear, and lung infections, and this includes Azobacter and Azomonas. So Pseudomonas has what's called a polar flagella. So see, you have that whole tuft of flagella on one end. That is very characteristic of Pseudomonas. The Delta proteobacteria, these include a wide variety of metabolic types, Desulfovibrio and Bdellovibrio, and the Myxobacteria. So here is a interesting specimen, the Bdellovibrio. This is a gram-negative pathogen of other gram-negative bacteria. So this is a bacteria that only infects other bacteria, and they all have to be gram-negative. This one is showing you the life cycle of Myxobacteria. So Myxobacteria are bacteria that infect fungi. Right. So anytime you see MYX, that usually tells you, or MYC, either one of those usually tell you that it has to do with a fungi. And then the class Epsilon Protobacteria, there's two important genera that we talk about here, the Campylobacter and the Heliobacter. The class Zeta Proteobacteria, these were first discovered based on their DNA sequences. The DNA of Zeta Proteobacteria are common in oceans. Only a few organisms have been cultured, and only one species has been formally named. Some other gram-negative bacteria you will want to be aware of are the chlamydias. These grow intracellularly, so inside of a cell, in mammals, birds, and some invertebrates. Some are smaller than viruses, and they are the most common sexually transmitted bacteria in the U.S. The spirochetes, these are motile bacteria that move in a corkscrew motion. They have diverse metabolism and habitats. Treponema and Borrelia both cause diseases in humans. The bacteroids, these inhibit the digestive tract of humans and animals, and there are some species that cause infections. And the cytophaga, these are aquatic gliding bacteria, and these are important in the degradation of raw sewage.